Tony, last time you were here was 4 years ago, I think, in Israel, and that's when we, Shay, you and myself, we decided to establish Precise. What we're witnessing is really a revolution, and this revolution has started decades ago, and really now is materializing. It's materializing thanks for Dr. Atala. We just heard the real history of what has been gone on in the last decades, and Professor Soker, we will hear, will be the next speaker. And not least by people like with the vision, as Mr. Maurice Kahn and Mr. Sami Sagol, we are honored to have them here in the audience. Collaboration between the industry and the academy is a powerful, proven concept. Academy brings it up to a certain stage, and the industry takes it forward from there and brings it to the market, and in our case, brings it to the clinicians and to the patients, as Tony said, really to improve their lives. Professor Tal Dvir and I have already discussed having a conference, the first International Regenerative Medicine Conference here in Israel next year. And we hope that it will happen here at Tel Aviv University. I have the honor to invite Professor Shay Soker from the Wake Forest Institute of Regenerative Medicine to talk about bioengineered tissue models of human development, drug metabolism, and disease. Dr. Shay Soker is a professor of regenerative medicine, cancer biology, physiology, pharmacology, and biomedical engineering at the Wake Forest Institute School of Medicine. Among his research contributions are the integration of molecular and cellular biology principles in regenerative medicine applications. Recently, Dr. Soker has applied his expertise to develop three-dimensional small tissue constructs that will allow for testing of new drugs without the need to test in animal and as models for disease such as cancer. This technology will allow to develop new drugs for treating cancer patients and also to test the effects of the treatments on cancer cells from each individual patient. Shay, please.
Toda Aryeh. That would be, it's a great pleasure see here friends and colleagues. And thanks for Aryeh and Tal for organizing it, and Maurice and Sagol family for being with us and supporting what we're doing. So Aryeh just said that there's a need for better systems for developing new drugs. And before I'll get into that, what I wanted to start with is saying that what you've seen, what you heard Tony talking about 25 years of experience in bioengineered tissues. What we can do also in the lab is take all these technologies and all this knowledge and actually put it towards other purposes, and one of which would be developing new drugs. And the problem is that currently there are two main systems for testing. One in cells in culture, and the other one is small animal models. And as good as they are, and most of the drugs that we're actually seeing nowadays have been developed and tested in these systems, they're not perfect. And they're not perfect because they take the tissue out of its context, and as you can see here. Each one of them has disadvantages, and then there was a great need to develop new systems. And actually, I decided that instead of me giving you the introduction to this field, if you care to see the full TED Talk of Dr. Francis Collins, the head of the NIH, I can just play a small clip because he does it better than, I guess, anyone else. No, yeah, it will. the speaker system doesn't work in the auditorium.
What's a drug? A drug is made up of a small molecule of hydrogen, carbon, oxygen, nitrogen, and a few other atoms, all cobbled together in a shape. And it's those shapes that determine whether, in fact, that particular drug is going to hit its target. Is it going to land where it's supposed to? So look at this picture here, a lot of shapes dancing around for you. Now, what you need to do if you're trying to develop a new treatment for autism or Alzheimer's disease or cancer is to find the right shape in that mix that will ultimately provide benefit and will be safe. And when you look at what happens to that pipeline, you start out maybe with thousands, tens of thousands of compounds. You weed down through various steps that cause many of these to fail. Ultimately, maybe you can run a clinical trial with 4 or 5 of these. And if all goes well, 14 years after you started, you'll get one approval. And it will cost you upwards of $1 billion for that one success. Wouldn't it be nice to be able to test a drug to see if it's effective and safe without having to put patients at risk? Because that first time, you're never quite sure. How do we know, for instance, whether drugs are safe before we give them to people? We test them on animals. And it's not all that reliable, and it's costly, and it's time-consuming. Suppose we could do this instead on human cells. You probably know, if you've been paying attention to some of the science literature, that you can now take a skin cell and encourage it to become a liver cell, or a heart cell, or a kidney cell, or a brain cell for any of us. So what if you use those cells as your test for whether a drug is going to work and whether it's going to be safe? And ultimately, because you can do this for the individual, we could even see this moving to the point where the ability to develop and test medicines will be you on a chip. What we're trying to say here is the individualizing of the process of developing drugs and testing their safety.
Okay, well, this was Francis Collins, and what Dr. Collins was describing is a body-on-a-chip system. And what I'm going to do here is describe to you the components of the system and what we can do, how are we using it. So the first unit that we engineered was liver. And you can see those livers are small. They're about 200 micrometer in size. And they contain all the major all the major cells in the liver. And then we go through, first we characterize the duration that these liver organoids are viable in the laboratory. You can see in the top right how the cells are actually organized. Within a few hours, you can get those organized, and those organoids are actually viable in the laboratory for more than a month. And they contain all the important cells, the hepatocytes, the biliary structures, endothelial cells, as well as cells from the immune system, the Kupffer cells. But the most important is the function. So are these organoids functions? And yes, they are. They actually they secrete urea and albumin and alpha-1 antitrypsin. So, but can they metabolize drugs? And actually, you can have several tests that would look at some of the CYPs of the CYP4150 isoforms, and you can see that yes, all the way up to 4 weeks, those enzymes are active. And importantly, you can see here is that the gray bars represent the hepatocytes in a 2-dimensional culture system. And in 2-dimensional culture system, within a week to 10 days, you lose all the activity. So this organization of a 3-dimensional organoids provide them all the signals that they need for long-term viability and function. So first drug that you want to test in a liver is acamol, Tylenol, and acetaminophen. And what you can see here that, as expected, with increasing amounts of the drug, there's a significant decrease in the production of albumin, which would represent the function, the decreased function of the organoids, and the same as urea. So why is this system important? The system is important because we want to use that system to develop countermeasures. So, for example, here you can use N-acetylcysteine. It's an oxygen scavenger, and N-acetylcysteine, actually, you can see here that would actually revert the effect of acetaminophen, and you can see that these organoids that have been treated with N-acetylcysteines have maintained a function for a longer duration. So that was a quick introduction into the first organoids, the liver. These are the heart organoids. So liver organoids were made all from primary cells. You can't really expand in culture primary cells from cardiac, so these were using induced pluripotential cells, IPS cells. And interestingly, when you get a collection of IPS cells, of cardiac differentiated IPS cells, they contained a proportion of different cells representing different cells from the heart. These cells are not only viable for long-term in the laboratory, but they can also beat. So in order to actually measure and quantify the beating, we have a special system where we have an LED light, and then below we have a camera. And one of our students developed a simple MATLAB script that can actually record the movement of these organoids and actually create a heartbeat, an EKG plot here. And this is the normal beats per minute. I think we have it at around 50 to 60 beats per minute. All right, so the next organ or organoid system that we worked on was the lung. And in the lung, there are different ways of creating those organoids. They can be planary, or they can be circular, spherical. In this case, we chose the planary one. And the main reason is that we wanted to look at the epithelial barrier of the lung and its function. So how do you create those lung organoids? You can see in the top left, you can see the structure of the lung. So we layer epithelial cells and the lung mesenchymal cells, as well as airway endothelial cells in a layer-by-layer fashion. And actually, you can get those organoids. You can see a very nice histology, very similar to the lung. And you can see expression of genes and functions of the lung. Another organ of great interest to our institute, our brain. So the main problems with brain that is impacted by drugs is the blood-brain barrier. So in this case, we couldn't really replicate in this system the blood-brain barrier of the brain capillaries. However, a group in the laboratory was able to create brain organoids that have been covered with endothelial cells. So now the endothelial cells are outside of the brain, but as you can see here, they maintain a very nice monolayer structure with expression of endothelial-specific markers, and maintenance, as I said, of a very nice monolayer. Whereas if you make it flat in a two-dimensional structure, they don't really make nice monolayer. So the effect of several cytokines, you can see here we started with histamine, which is probably the strongest vasodilators, and you can see perfusion of a fluorescent green dye into the liver organoids. So the system is responding to signals. And then we ran a test of multiple cytokines here, and you can see The Z01 is actually marking the boundaries of the endothelial cells, and with IL-6, TNF-alpha, and just with low oxygen, you can see a complete disruption of the endothelial monolayer outside the brain, which suggests that now the blood-brain barrier is completely destroyed. The last organoid system that we have created in the laboratory are testes, and we have a group in the laboratory that are interested in using testes for preservation of sperm stem cells. So we acquired that technology from that group. And in the testes, you have three basic cell types that you need to maintain: the spermatogonial stem cells, Sertoli cells, and Leydig cells. And you can see here they're organized in a spherical way, where the Leydig cells are on the outside, and the Sertoli and spermatogonial cells are kept in the inside. So why are testis organoids important? We know that there's a decrease in fertility in the Western world. And some of the reasons are environmental, or drugs, or pesticides, and so on. So we've actually looked at some drugs and their effect on viability of these organoids. So I don't want to go into details with the effect of different drugs, but just to show you that they all have a different effects on the viability. And if you compare this activity to monocultures of each of the cell types that are contained in those organoids, they would have no effect. So if these drugs were tested for toxicity or for effect on fertility, no one would know that they would have effect. However, if you take all the cells together in a three-dimensional organoids, now you can see their effect. As I said before, environmental toxins are very much in discussion in recent years. So we've tested mercury, thallium, and lead, and you can see here the huge difference between the IC50, the amount that is required to decrease by 50% the viability of the cells in a two-dimensional culture versus a three-dimensional culture, and you can see the great difference. So in the two-dimensional, there's a direct exposure of the toxin to the cells. It's very toxic. It's not so toxic when you talk about a three-dimensional structure. So these were each of these organoid systems by itself. However, it's very important actually to put them together and how they work in concert, like in our body. Are they going to work as individuals, or they're going to have a cross-organ effects? And so to do that, actually build a system. It's a fairly primitive system. This is prototype 1. We now have much better developed prototypes. And in the first system, we tested a two-organ system, liver and cardiac. And basically, there are two chambers, and they're connected. We can bypass one, and then we can test the effect of different drugs. And the first drugs that we tested were epinephrine. So epinephrine is a ligand for beta-androgenic receptor, and it would speed up heartbeat. The other drug is propranolol, and propranolol is a non-selective beta blocker, and propranolol will antagonize the effect of epinephrine and would slow it down. So let's see what happens now if we put these two drugs in the system. And kind of long story short, and we'll go from top to down, if you start from baseline, so this is the heartbeat per minute, you can see that's the normal. If you put epinephrine, heartbeat goes up, so you can see more peaks. If you put propranolol, then it goes down to more or less the same as control. But what happens if you first blow the propranolol through the liver. The liver would metabolize the propranolol, and then it would have no effect on the epinephrine. So that's very important because now we can actually see the interaction between different organs in the body. And why is it important for some drugs to be metabolized in the liver, whereas others should not get into the liver because they would either block their activity or make them toxic. And this is the graph, the summary. So from the 2-organoid system, we actually went into the 3-organoid system. Now we've introduced the lung into liver and heart. And so bleomycin is antibiotics, and it's known to have an effect on the lung. So you can see this is viability. Green means cells are alive. Red means cells are dead. You can see no drug control. Many cells are alive in the liver and the cardiac. But when we introduce bleomycin, there's a significant decrease in cell viability. So we did not see much effect on the liver, on the lung and the cardiac, and it was a little strange because we were expecting it to have. So we asked the question, we now separated the system, and we asked the question, how can it be? So if you have no drug control, this is heartbeat per minute, it's about 33. If you have bleomycin in the 3-organoid system, now the heart doesn't beat. However, if you have bleomycin on the heart only, now the heart still beats. So it's a little confusing. How come the bleomycin in the 3-organoid system has such a determinal, such a harsh effect on the heart, whether in the directly it does not? And what it turned out is that I'll give you the solution already. It turns out that by exposing the lung to bleomycin, you get expression of IL-8 and IL-beta and IL-1 beta. And you can see here that these are the curves where if you have bleomycin, you have an increasing in both of these cytokines. And what happens is that those cytokines are actually cardiac toxic. So again, showing you the importance of having multiple organoids connected in one system and the flow of the drug from one organoid to the other. Because again, exposing of the lung to bleomycin causes secretion of cytokines, and those cytokines are now cardiotoxic. So if the drug was tested only on heart cells, it would show no effect, no toxicity, but in patients, it did. And then just a small proof that you can have many organoid types in one system. This took a long time to establish because each of these cell types require different media. And now they have to compromise on the conditions of the media. But now we can actually have, and this paper was published just recently, 6 different organoid types in the same system. So this one was just to show that if you take a chemotherapy like capecitabine and you run it through the system, starting with the liver, cardiac, lung, testis, you can see cardiac toxicity, you can see lung toxicity. However, if you remove the liver, there's no toxicity. And the reason is that the drug is being metabolized into toxic residues, into toxic metabolites, and then that is toxic to the downstream organs. So up to now, I sort of described to you some of the uses of this organoid system to test drugs and test the potential toxicity of drugs for drug development. But more importantly, can we use this organoid system as a disease model? Because you can have an animal disease model, but animals are not small humans. Mice are not small humans, and those knockout models in mice don't always replicate all the intricate tissue components of human. So can we create now diseased organoids that will then teach us a little bit more about the disease and would allow us to have a platform for treatment for these diseases? And the first disease that we addressed was liver fibrosis. This is not a liver fibrosis talk, so I won't go into all the molecular mechanism. Just to say that in liver fibrosis, there's a significant expansion of some cell types in the liver. Those are liver stellate cells. And those cells are acting like fibroblasts. They secrete a lot of collagen, and they make fibrotic tissue. So what we done in the lab, set up a system where we combine liver progenitor cells, the HepRGs, along with collagen and the hepatic stellate cells. And what you can see here below, you can see that this is contraction of the collagen. As the more contracted the collagen, the more fibrotic, the more stiff it is. And you can see that we have two types of stellate cells, non-activated, the LX2, and the activated hepatic stellate cells. And the activated stellate cells are contracting a lot more than the others. We can actually quantify the level of collagen remodeling, how larger and wider the collagen fibers, which is a hallmark of fibrosis, are in each of these organoids. So the more red, the more remote, the more bundled collagens are, and you can see the same difference. So the shrinkage in size and the increase of stiffness is actually is causing by rebundling of these collagen fibers. So that's good. So we have a fibrotic model, but what effect this sort of fibrosis would have on liver development? So we've actually looked at biliary duct malformation, and Tony had shown you our model of fetal liver. And again, I won't go into the disease too much. It's just that during development, the biliary ducts are formed, and in case of liver fibrosis, they're abnormal. They don't form normally, or they don't form at all. And so here's what you can see. So these are the biliary duct structures. They're stained in red here. And what we could see is that when we use the unactivated stellate cells, they are required for making of these tubular structure of the biliary ducts. But if you have the activated ones, there's a significant decrease in the number of functional or lumen containing ducts. So again, now we have a model that would simulate a fetal biliary duct formation. Another model in these organoids are effects of drugs on liver fibrosis. This is triglyceride, a drug commonly used as an anti-inflammatory. And the point here is just to show you the effect on viability of liver cells that are in two-dimensional structures versus the organoids. In two-dimensional structured cultures, it's a straight line. It's that sort of green line, and there's hardly any effect, whereas there's a lot of effect in the organoids. Now, what triglyceride might do is actually induce a fatty liver, liver cirrhosis. steatosis, and you can see here accumulation of fat and phospholipids in these liver organoids. So there are two ways of inducing a disease. You can do it by pharmacological agents, by drugs, or you can do it by activating or changing the cellular milieu in these organoids. This is, again, I mean, a very known case about, I think it's close to 10 years ago that COX inhibitors were pulled out of the market. These drugs were blockbusters, and then they were actually pulled for several years from the market. Now they're back again with big warning because they cause cardiotoxicity. That cardiotoxicity was not discovered in cell cultures, was not discovered in mice. But we can see here that had we had that technology when COX inhibitors were developed, they may have seen that this drug has some effect on cardiac cell viability. Just another model of inducing cardiac toxicity. You can use cardiac toxicity just by actually exposing those cardiac organoids to different drugs. Angiotensin, again, used by many patients. And here we know that it increases blood pressure. We can see that it increases heartbeat. Inducing of fibrosis with TGF-beta. So now we have sort of a collection of reagents that can induce a disease state in these organoids. So lung is also of a great interest for us, both because we are engineering lungs for use in patients, but also because the lung represents one of the entry of many drugs, all the aerosol drugs and so on. For lung researchers, cystic fibrosis is probably the most important lung disease. So what we've done here is actually use the CFTR172, which is an inhibitor of the receptor, and that's what actually happens in cystic fibrosis. We can see here, this is a measurement of the barrier function of the lung. You can see in that sort of middle picture, this is the system. What we're recording here is the barrier function. You can see when you put amiloride, immediately we have a drop in the barrier function, and then when you put forskolin, it goes up. However, you can see a significant difference whether we have the three-dimensional organoids or just a two-dimensional lung basal epithelial cells. They don't really respond the way as the organoids, and they would not replicate the lung barrier that one might see, lung barrier function that one might see in cystic fibrosis. Lung infection is also a very important aspect, and we can see here that our lung organoids can actually show binding of, in this case, these are fluorescent bacteria, and we can see that the amount of infection in those three-dimensional organoids is much higher than in the two-dimensional culture. So in the last maybe 10 minutes, I want to go into one of our major efforts in developing disease models in these organoids, and this is cancer. So cancer research has been around for many years, and one of the problems that we can't really get new drugs is that most of the drugs, anti-cancer drugs, are tested on either animal models or on individual cells in cell cultures. And so they lack all the microenvironment around them, which now is well understood supporting the tumor growth. So I'll just show you a few examples. This is an example of a metastasis-on-a-chip model. So we chose the colon cancer metastasis to the liver here. So in one chamber, you have the gut organoids, And in the other system, in the other chamber, you have the liver organoids. And this system allows the cells to freely actually flow from the gut to the liver if they metastasize. In order to quantify it, the tumor cells are going to be labeled in red, and we have a small, actually it's a small camera that was taken from an old iPhone that can actually record the fluorescent cells. So this is what we're seeing. So now we're looking at the gut organoid, and you can see over about 14 days that the cells, the red cells, the red metastatic colon carcinoma cells are growing inside the organoids, and by about day 14, they start to actually disseminate into the medium. So remember day 14, okay? And these are the liver organoids. And the liver organoids, what you can see is a streak of red cells that actually is penetrating or now are disseminating into the liver. So in the experiment that we've done, we took two different colocarcinoma cell lines. The black ones are highly metastatic, and the blue ones, the blue line, represent a low metastatic. And as you can see, both of them in the primary site, in the gut, they actually grow very well, and they reach a plateau after about 14 days. But now look at the right side of the graph, where now we're recording the amount of cells in the liver organoids. And what you can see is that at about day 14, now we start seeing some cells in the liver organoid, and those cells keep on increasing in their fluorescence intensity, whereas the non-metastatic cell line doesn't even reach to the liver. So that's one example of how can you use the organoid system now to look at metastases. Now, the most important use of these systems are to incorporate patient-owned cancer cells and to test them directly for drugs or for other manipulations. So that's a new variation of our organ-on-a-chip system, where we can actually have multiple organoid system in one microfluidic. And we've done now about 10 different types of cancers. This one is mesothelioma. Mesothelioma is a cancer in the lung caused by asbestos. It's a non-treatable cancer, but right now it's being treated with different chemotherapies. And the main problem for the oncologist is what drug to use, because this, as I said, there's no cure. So there are two patients there, and you can see the difference in their response. And again, the response would be cells that are dying, that are responding to the drug, are going to show as red, and cells that do not are going to show in green. So you can see that in patient 2, in general, with the different cocktails of chemotherapy, most cells are green. So this patient, spare this patient from this chemotherapy. This patient is probably not going to respond, whereas patient 1 responded very nicely to the cocktail. But even further, now many of these patients, their tumor cells are being sequenced in order to analyze mutations that you can actually target with specific drugs. So in this case, one patient 2 from this other study actually found to have a mutation in the EZH2 gene. So, I'm sorry, has an elevated EZH2 gene. And let's not go too much into the function of EZH2, but there's an inhibitor for EZH2. So in this organoid system, we actually tested these two patients for the DZNEP, the EZH2 inhibitors. And you can see that patient 1, which is in blue here, did not respond, but a patient 2 actually responded much better to the, I'm sorry, patient 1 that did not respond to the chemotherapy cocktail now is responding to the EZH2 inhibitor. So this is a very powerful system to actually pre-screen for drugs that that patient might respond or not respond to. And I'd like to end with, the recent technology that we've tested in these organoids, and this is immunotherapy. So immunotherapy has come to the forefront for treatment of many cancers. It is based on the fact that T cells, when they interact with an antigen, they start proliferating, but they would not proliferate forever, and because they need some mechanism to hold them in place so that they won't kill the entire individual. So these are called checkpoints that cause the T cells to stay and not proliferate and not exert more killing. So problem is that tumors had actually found out about it. And what they do, actually, they signal the T cells, well, stand in your place, stop proliferating, stop killing us. And so the immunotherapy that works here is to bypass the checkpoint that the tumor actually imposes on the T cells. And that's the reason why we call them checkpoint inhibitors. So we actually used the first system is a system in a mouse. The MC38 cell line is a T cells that are not dependent on those sort of checkpoint inhibitors. And you can see that when T cells are present, they will kill the cells in the organoids. However, the CD26 is dependent on the treatment, and you can see it's only when we apply the cocktail of checkpoint inhibitors, PD-1, anti-PD-1, and anti-CTL4, now we can see killing. And we've actually taken it to humans, and you can see here these are the two drugs, the two leading immunotherapy drugs. And you can see that if we include cells from the lymph node in these patient-specific tumor organoids, we can see killing more red cells, whereas tumor organoids alone are not. So let me just conclude by saying those organoids can stimulate organ development and congenital diseases, as I showed you. Compared with culture cells, two-dimensional culture cells, organoid better resemble a tumor in their natural tissue environment. And basically, going down to the last bullet point, those bioengineered tumor organoids technology is a platform for not only testing current drugs, but also to develop new ones. So with that, I'd like to thank the people that had actually contributed a lot to this work. You know the guy in the top right panel there. Tony, who had actually led and is leading the Body-on-a-Chip program at WFirm. Tom Shoup is working with Tony on that program. Alex Cardall works with me on the tumor organoid system. Sean Murphy develops the lung organoids. Goodwell had developed the brain organoids. Colin Bishop is in charge of the liver and cardiac organoids. And Human worked on the testis organoids. And the others are students and postdocs that worked with me. And this has not been possible without the support of funding agencies. So thank you. And if we have any time for questions, I'll be happy.
Thank you, Shay. We have time for one quick question.
The Wnt signaling, we have not looked at that. But if you're talking about the Wnt signaling for differentiation of these organoids, we have done it in the fetal organoid system, and they do respond. We've actually worked mostly on the Wnt signaling in the cancer organoids. So I'll go to the last question because I still remember it, whether they maintain the cancer stem cell phenotype. We have not directly looked at that, but what we've seen is that actually what we're most interested in this case is the clonality. So this is a different aspect to your questions. And what we're seeing here that we preserve clonality much more than in a 2D culture. Because when we do a single cell genomics on cells that we get from these organoids, we get a much wider pattern or representation of the clones that we get in the original biopsy. So we assume that they will also maintain the stem cell phenotype, but we've not directly tested that. So if you remind me the second question, then I can quickly maybe answer that. Right. Correct. So by placing them actually in a three-dimensional structure along with other cells, we actually see very little proliferation. There's a feedback inhibition, so there's maintenance of cells. Cells are viable, but there's very little proliferation. We can talk later about the tumor organoids. In the tumor organoids, we do see cycling, so proliferation, death under some conditions.
And before we conclude, I just want to announce that next week we will have Professor Robert Langer from MIT here to give a talk. And with that, I would like to thank Shay and Tony again for their great